Hey guys, so we are at the second video of the second chapter of class 12th NCERT, right, which is sexual reproduction in flowering plants. So in the last video, we discussed what? We discussed what is the, a section of a flower, what all structures it has. And then we started off with the structure of the male reproductive part of the flower, which is the stamen. So we saw that stamen has anther and filament basically is attached with the petal or thalamus of the flower, and then anther basically has the pollen grains, which helps in fertilization, which contains the male gamete, right? So anther basically is nothing but a bilobed structure, and each lobe is dithecous, right? And anther is basically a tetragonal structure which contains of four microsporangia, right? And these microsporangia then basically become pollen sac, and they have pollen grains. They are basically packed with pollen grains, right? So then we saw the structure of the microsporangium as well, how it is exactly four wall layered, and the four layers are basically epidermis, endothelium, middle layers, and tapetum. And basically the first three layers, what do they do exactly? They help in protection and dehiscence of anther to release the pollen. And then the innermost layer, which is tapetum, it nourishes the developing pollen grains, right? And then we saw that they have sporogenous tissue as well, which is exactly the center of the microsporangium when the anther is young, right? Now, what exactly is microsporogenesis? So, we are going to cover microsporogenesis first, and then we'll see the structure of a pollen grain as well, right? So, as the anther develops, what happens is the cells of sporogenous tissue, they undergo meiotic division and they form microspore tetrads, right? So each cell of this sporogenous tissue is basically capable of giving rise to a microspore tetrad, right? Is each cell, right? So each one basically is a potential pollen or a microspore mother cell. And even in the previous structure, in the previous video, we saw microspore mother cells as well, right? So each one, basically each of the sporogenous tissue is capable to give rise to a microspore tetrad, and each one can be a potential pollen or microspore mother cell, right? So basically the process of formation of this microspore from a pollen mother cell through meiosis is known as microsporogenesis. So what exactly is microsporogenesis? That when these microspores are formed from the pollen mother cell through meiosis, then it is microsporogenesis, right? So microspores, when they are formed, they are basically arranged in cluster of four cells, and it is known as microspore tetrad. It is known as microspore tetrad. Okay. Now, as the anther matures, always remember that as the anther matures and dehydrate, what happens? The microspores dissociate from each other and they develop into pollen grain. They develop into what? They develop into pollen grain. And inside each microsporangium, there can be several thousands of microspores or pollen grains, which are then released with the dehiscence of anther, right? So if you see this even structure of a pollen grain, what you see, this is a mature dehisced anther. This is nothing but a mature dehisced anther. So what we saw first, that it is a tetrad. So you can see that there are four lobes basically, right? It is tetragonal in structure, and then it has microspore mother cells inside the four layers, right? And when this matures and dehisces, then the pollen grains. So these are nothing but the pollen grains, and these are released outside in the environment basically. right? So basically, what exactly is the pollen grain, right? So let's see what exactly is the pollen grain. Pollen grain basically represents the male gametophyte, right? So it represents the male gametophyte. It is usually spherical in shape, okay? It is usually spherical in shape and measures around 25 to 50 micrometer in diameter. So remember that it measures around 25 to 50 micrometer in diameter. Okay, so if you ever touch and open anthers of hibiscus plant or any flower that you can find where you have even in lilies you'll find yellowish powdery pollen grains on your finger once you touch the anther, right? So if you ever want to see the structure of that pollen grain, just sprinkle these grains on a drop of water, okay, on a glass slide, and then if you observe it under a microscope, you will find the exact structure of that pollen grain, right? So it is basically very different in different species. Now basically in general they are spherical in shape and around 25 to 50 micrometers in diameter. Okay, now they basically have prominent two-layered wall. Okay, the outer covering is hard and is known as exine, and the inner covering is basically the intine. Right? Now, how exactly is the exine? So, exine is made of sporopollenin. Remember that exine is made of sporopollenin. Okay, so if I tell you the exact structure, it will be like this. Mm. This is nothing but a tetrad. This is a primary cell wall. There's a callose layer, and then inside there are microspores. Okay, so inside is the pollen grain, right? Now, this pollen grain has a hard outer covering, which is exine, which is made up of sporopollenin. Now, sporopollenin is known to be one of the most resistant organic material. Okay, remember that it is known to be one of the most resistant organic material, right? Now, basically, why this layer is so hard and why it is so resistant, it's because it can withstand high temperature and strong acids and alkalis, right? So it is basically for protection. So no enzyme that degrades sporopollenin is so far known. Remember that there are no enzymes known which can degrade sporopollenin. Okay, and pollen grain exine has prominent apertures which are known as germ pores, right? And this is the point where sporopollenin is absent. So basically, pollen grains are well preserved as fossils because of the presence of sporopollenin, right? And exine exhibits a fascinating array of patterns and designs as well, right? So it it can be like this. So this is a pollen grain and there will be hard outer covering of say this shape. Okay, so this layer is of sporopollenin and this is the exine layer basically. Okay, now what about the inner wall? So the inner wall of the pollen grain is basically called intine. Remember that it is very different from exine as it is a thin continuous layer which is made of cellulose and pectin. Okay, so it is made up of cellulose and pectin. Now the cytoplasm of pollen grain is basically surrounded by a plasma membrane. Okay, and when the pollen grain is mature, it contains two cells. Okay, so the one is the vegetative cell and the other one is the generative cell, right? Now, this vegetative cell basically is bigger, it has abundant food reserve, and it is irregular in shape with respect to the nucleus. So it has a very large, irregularly shaped nucleus. Okay. The other one is the generative cell. Now, the generative cell basically is small and it floats in the cytoplasm of the vegetative cell, and it has simple spindle shaped with dense cytoplasm and a small nucleus. So it is spindle shaped, dense cytoplasm and a nucleus, right? So basically in over 60% of angiosperms, the pollen grains are shed in two-celled stage. Okay, and in the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed, which is a three-celled stage. So if I show you the diagram, it is nothing but this. Okay, so basically this is a wait, this is a pollen grain. Okay, and then it basically is maturing, and then it has one nucleus and it has various vacuoles. Okay, then you'll see that it is forming an asymmetric spindle, and then it matures into a pollen grain wherein it has vegetative cell and generative cell, right? And later on, this generative cell, so basically this is the two-celled stage. But later on, what happens? That this generative cells that divides and gives rise to two male gametes in some cases, which is a three-celled stage. Right. So this basically is nothing but structure of a pollen grain. Now these pollen grains in many species, they cause severe allergies and bronchial infections as well. Like it even causes asthma in many animals, or even bronchitis, etc. Right. So they are very much rich in nutrients, and they are sometimes used as food supplements as well. So it's not just the male reproductive organ, or the organ which is basically carrying the male gamete. It also is useful in various ways. So since they are rich in nutrients, it has basically become a source of food supplement as well, right? Now, in many countries, in many Western countries, a large number of pollen products in the form of tablets and syrups are available in the market, right? So basically, it even is said to, increase the performance of athletes or race horses, right? Now, basically, pollen grain of rice and wheat. So they basically, what happens is lose viability within 30 minutes of their release. So now different pollen grains of different plants basically have their own life cycle, right? So in many plants like rice and wheat, they lose viability within 30 minutes of their release. While in some plants like many, leguminous plants or rose varieties, it can be viable for months, right? But then in some plants, it's even possible to keep them for years, right? So what happens is that in case of plants as well, there are pollen banks formed, or which is similar to basically seed banks, something like seed banks, wherein you store and preserve pollens, and you at the basically time of proper fertilization, when the conditions are fine and when the plant can basically be germinated, you use that pollen grains by artificial and artificially transfer it to the you transfer it artificially to the plant, and you make them fertilize basically when the conditions are favorable, right? These basically are the pollen grains, which are nothing but a two-celled stage wherein it has one generative and one vegetative cell, right? And that generative cell is nothing but the male gamete that forms the male gamete, right? So here we finish the microsporogenesis and the pollen grain. Now in the next video we'll see the female uh, reproductive part, which is the pistil. Then we'll see what is the megasporangium and what exactly is the embryo sac which contains the egg. Okay, so I hope you understood what exactly microsporogenesis is, and what exactly is a pollen grain, what is uh, its characteristic features, and what are the various uses of pollen grain, apart from the normal uh, use of it in fertilization in plants, right? So if you did not understand any part of today's video, please do let me know in the comment section or on the WhatsApp number given in the description of the video. And yes, if you did, do not forget to share it with your friends, and do not forget to give it a like, and please subscribe to the channel as well so you are updated with all the videos that I'll be putting up, right? Okay, guys, thank you. Bye bye.